Good morning, everyone. Today we will discover the power of AptPlex kit, which can be considered as the next generation panel solution. So let's begin with today's roadmap about what we'll discuss. I will introduce what is multiplexing and how it can be used in detection of cytokines, chemokines, and other growth factors. Then we'll learn about AptPlex manufactured by eLab, its working principle, its technology, and afterwards I will also have a brief look on to its components and advantages, and lastly, really brief look on the competitive analysis with the other brands. Okay, so let's learn about multiplexing, but before that, we should know why do we need this technology actually? Cytokines, including interleukins, tumor necrosis factors, and interferons, serve as central regulators for immune communication. They control the immune activation, suppression, and coordination by binding to specific receptors and initiating the intracellular signaling pathways. Interleukins regulate differentiation of T helper subsets. Then TNF are the key mediators of inflammation and programmed cell death. And interferons, these are essential for antiviral defense and immune modulation. As we know that chemokine provides a directional control of immune cell trafficking. By forming a concentration gradient, they guide the immune cells to a specific site, such as infection, inflammation, or tissue injury. This ensures the spatial precision in immune responses and enable efficient immune surveillance and recruitment. If we talk about growth factors such as VEGF and CSF, they regulate the tissue repair and cell production. VEGF promotes angiogenesis to restore oxygen and nutrient supply in damaged tissues, while CSF that stimulates the granulocyte production and release from the bone marrow, linking the immune activity with the regeneration. Soluble receptors and adhesion molecules, they reflect the cellular communication and migration processes. Molecules such as CD25, they indicate the immune activation status, while ICM1 or VCAM1, they regulate the adhesion and transmigration across endothelial barriers. These markers are widely used in assessing the inflammation and immune cell infiltration. These mediators actually operate through overlapping the signaling pathways. It's like seeing the immune system in action, moving, talking, and coordinating all at once. Immune signaling mainly involves multiple mediators acting in parallel rather than isolated pathways. The balance between the activation and regulation determines whether the immune responses remain controlled or become dysregulated. Under normal conditions, cytokines and chemokines signaling support regulated immune activation, targeted cell migration, and resolution of inflammatory responses. Growth factor activity contributes to tissue repair and restoration during the immune responses. Then we have some alteration in these signaling pathways or signaling patterns that are associated with different disease states. Excessive signaling activity is linked to hyperinflammatory conditions such as cytokine storm, while reduced or impaired signaling can weaken the immune defense and increase infection susceptibility. Changes in the receptor and adhesion molecule expressions are observed in autoimmune conditions and tumor progression, affecting immune cell behavior and even its localization. So these signaling pathways are commonly targeted in therapeutic strategies through inhibition of pro-inflammatory mediators, receptor blockade, or modulation of immune activation pathways. Multiplexing actually allows simultaneous measurement of these multiple analytes, which we have just discussed, within a single assay format, replacing the need for multiple individual tests like ELISA. This reduces the experimental repetition and increase the amount of biological information obtained from each sample. By capturing several immune mediators in parallel, it enables a more complete representation of immune signaling activity, including interactions across cytokines, chemokines, growth factors, and soluble receptors within same experimental context. So it's literally a game changer. The approach supports the biomarker discovery by enabling the comparison of multiple candidate molecules under identical conditions, improving the consistency and identifying disease-associated expression patterns. This is particularly useful in complex conditions where single marker analysis is sufficient. Sample composition is reduced because multiple targets are measured from the same limited volume, which is critical when working with clinical samples, pediatric samples, or rare experimental material. Assay performance is also enhanced through improved sensitivity and specificity, and along with the broader dynamic range that also accommodates both low and high abundance analytes in a single run. So you can see where it goes. Then we also have the operational efficiency improved by reducing the assay number, reagent use, and instrument time, which directly impacts the experimental throughput and workflow scalability. The global multiplex detection immunoassay market is rapidly expanding, reaching about $3.2 billion in 2025 and projected to grow to $4.4 billion by 2032, with a cumulative annual growth rate of 4.4%. In research, these tools are transforming how we study the complex biological systems by allowing multiple biomarkers to be measured simultaneously. This boosts the efficiency, reduces the sample volume, and produces more complete picture of immune signaling and disease models. Advances in multiplex ELISA, fluorescence-based assays, and high precision detection are making these studies more sensitive and reproducible. Okay, if we talk about the traditional ELISA, those assays were based on workflows measuring just one analyte per reaction, which means each biomarker requires a separate assay step. This increases the number of plates, number of reagents, amount of reagents, experimental steps required for a single study. In many biological samples, especially the clinical or primary specimens, the available volume is always limited, like in mouse samples. Then running a multiple single assay quickly becomes restrictive because each target consumes additional sample. Immune processes do not occur in isolation. Cytokines, chemokines, growth factors, and soluble receptors change together within the same biological environment. Measuring them separately does not capture these parallel changes under identical conditions. Separate assays also introduces the differences in incubation, handling, and processing, which leads to variation between the measurements when the results are combined later. Multiplex formats allow multiple analytes to be measured within the same reaction setup. A single sample is sufficient to generate a broader data set without repeated processing steps. This approach reduces repeated freeze-thaw handling, which helps maintain sample integrity, particularly in the stored or precious biological materials. It also allows better use of limited sample input to study designs where multiple time points or multiple markers need to be evaluated from the same source. What are the main advantages of multiplex? As we have just mentioned few of them, but there are more advantages, like if we are measuring the same reaction environment instead of running the separate tests for each target. One sample generates multiple readouts in the single workflow. This creates a higher density of biological information from the same input material. The data reflects multiple components of immune signaling within the same experimental context, which enables a simultaneous observation of cytokines, chemokines, and other soluble factors, providing more connected view of signaling activity across different pathways. When multiple candidate biomarkers are analyzed together under the identical conditions, comparison becomes more consistent and suitable for identifying the patterns which are associated with disease states. The detection system is also designed to cover a wide dynamic range, allowing both low abundance and high abundance analytes to be measured within the same run without losing the resolution. Analytical sensitivity and specificity are also maintained while handling multiple targets at once, even in the complex biological matrices. Workflow requirements are reduced because fewer separate assays are needed, which are also lowering the reagent consumption and instrument usage across different experiments. Now, why ELISA is not suitable for some kind of studies? Because ELISA measures one analyte per assay, requiring separate wells or plates for each biomarker. Each target is processed independently. Multiplex assay measure multiple analytes in the same reaction environment where all targets are analyzed together under identical conditions. Sample uses differs because ELISA distributes sample across multiple separate assays, while multiplexing uses single sample for multiple measurements. The workflow in ELISA always involved repeated setup and a repetition of steps for each analyte. Multiplex assay workflows consolidate these into one experimental run. Because ELISA measurements are performed separately, variations can occur between the assays. Multiplex assays reduce this by making all the analytes within the same reaction conditions. So data from ELISA represents individual markers independently, while multiplex assays generate a combined profile of multiple immune-related molecules from the same sample context. If we talk about several application, multiplex bead technology is being used across many research areas where multiple biomarkers need to be measured in the same sample. For example, in immunology, it is commonly used to measure the cytokines such as IL-6, TNF-alpha, IL-10, and IFN gamma. These are the most common cytokines, and in the same serum or cell culture sample or study inflammatory responses. In case of cancer research, it can be used to analyze tumor-related markers such as VEGF, EGF, and PDGF, along with inflammatory cytokines to understand tumor microenvironment signaling. In case of vaccine studies, it is applied to measure antibody-related responses and immune mediators like IL-2, IL-4, and IFN gamma after immunization to evaluate both humoral and cellular immunity. In the transplantation research, it is used to monitor the rejection-associated markers such as IL-2 receptor, TNF-alpha, and IL-17 in patient serum over time. If we talk about the infectious diseases, it also helps to track the host response markers like IL-6, IL-8, MCP-1, and CRP-related inflammatory signals during the bacterial or viral infections. Metabolic research also can't be denied. It is used, this multiplex techniques can also be used in the metabolic research to analyze the adipokines and metabolic regulators such as leptin, adiponectin, and insulin, and resistance in obesity or diabetes models. And last but not the least, neurobiology. It can measure neuroinflammatory markers like IL-1 beta, TNF alpha, BDNF, and in the brain tissues or cerebrospinal fluid. And you will be surprised, even this technology can be used in environmental studies. It can be used to detect the multiple toxins or stress-related biomarkers in water, soil samples, or in plants in a single run. Okay, why researchers use multiplex bead assays? Because these are commonly used in biomarker screening experiments where a larger set of candidate molecules is evaluated in the single run to identify the potential targets. They are also applied during experimental validation stages where the selected markers from an initial screen are confirmed across additional samples or conditions. In the comparative study designs, they are used to analyze differences across multiple conditions such as treatment groups, dosage variations, or genetic backgrounds within the same experimental setup. They are frequently used for pre and post intervention analysis as well, where the samples are collected before and after stimulus, treatment, or disease progression are assessed side by side. In the high throughput workflows, multiple samples are processed in batches to generate consistent multi-analyte data sets under standardized conditions. So it is also very important to use in these kind of workflows. Then they are also used in time course or kinetic studies, where samples collected at different time points are analyzed to observe the dynamic biological changes across same experimental system. Okay, then the most important factor for the multiplexing is to study it in mouse models. For example, Th1 and Th2, they represent two major arms of adaptive immune system, and their balance is completely assessed in mouse models to understand immune polarization under controlled experimental conditions. In mice, Th1 responses are associated with cellular immunity, involving the clearance of intracellular pathogens such as viruses and certain bacteria, and are also linked to tumor immunity and autoimmune related mechanisms. Th2 responses are associated with humoral immunity, which is important for defense against the extracellular parasites and is also strongly involved in allergic responses such as asthma or dermatitis. This balance is defined by cytokine profiles where Th cells are characterized by IFN gamma, IL-2, and where Th2 cells are associated with IL-4, IL-6, and IL-10. In the mouse experimental system, these markers are measured to determine how immune responses shift under different disease models or treatments and autoimmune disease models to evaluate the immune directionality and response patterns. It is also used in mouse vaccine studies to determine whether the induced immune response in Th1 dominant or Th2 dominant. If we talk about the preclinical safety studies using mice, it is also used to detect the abnormal immune activation or hypersensitivity responses triggered by these compounds. Okay, now we have came to at the end of our first section. Let's move on to the next section and let me help you meet our AptPlex kit. Okay, first of all, let's take a closer look at APT Plex, human or mouse TH1, TH2, 7-plex panel kit here. These ELAB Science APT Plex kits allows you to measure multiple cytokines, chemokines, and growth factors in a single sample, giving a comprehensive view of immune signaling. They are ideal for the studies in immune regulation, inflammatory responses, and tumor metastasis. Compared with the traditional ELISA kits, APT-Plex offer higher sensitivity and wider detection range. Currently, we have launched the human kits in the international market, and we will soon introduce the mouse panels as well. This kit will work with the cell culture supernatants, EDTA plasma, or serum samples, making them versatile and powerful tools for research. This kit comes with all essential components you need to run a multiplex assay efficiently. In case of premixed antibody conjugated beads, these can be used for the target capture. Then you will have the biotin-related detection antibodies and SAPE for signal development. The kit also provides the lyophilized standards for quantification, assay and wash buffers, and even the plate sealing films. Everything needs to be stored at 2 to 8 degrees with light sensitive components protected from light, and a detailed manual is also included to guide you through the procedure. These components together provide a complete ready to use solution for accurate reproducible multiplex analysis. If it is difficult for you to understand this kit, we are always here to help you out, understand more deeply about the components and how the operational procedure works and how to read out the data. So you can reach out to us anytime. Okay, in addition to the kit components, you will need a few standard laboratory items to run the assay. And these are the most common instruments in the research lab, so most users should have access to them. For example, U-bottom 96 well plates, a vertex mixture, and an incubator suitable for 96 well plates. You will also need a magnetic separator to handle the bead-based reactions, or if you have the magnetic rack, you can also use that one. We do not recommend you using the centrifuge machine because this technology is magnetic bead based. So it is very simple to use when you have the magnetic separator or the magnetic rack. And these are the simple equipments, simple plates or simple racks, which are available mostly in all the labs. Then you also have to need a flow cytometer equipped with PE, APC, and APC-CY7 detection channels to read the signals. And this kind of flow cytometer is also available in most labs, so you won't be having a problem in that. And now the principle. APT-Plex kit is based on the sandwich immunoassay principle, which is most similar principle as ELISA. Using the color fluorescence coded magnetic beads. In case of ELISA, we use the plate to coat the capture antibody, but here in this case, each bead is coupled with a specific antibody to selectively capture its target molecule from the sample. Detection antibodies labeled with fluorescent or biotin bind the capture targets to generate the measurable signal. For biotin-labeled antibodies, PE streptavidin is used to amplify the signal. This multiplex bead design allows simultaneously high-sensitive quantification of multiple analytes in single sample, providing a broad dynamic range and reliable results. If you don't understand the principle, let me explain in the easy words. This kit works on the sandwich immunoassay principle. Imagine each tiny bead as a fishing hook designed to catch one specific molecule from your sample. Each bead has a special antibody on its surface that binds only to its target. So the hook is just taking out that target. Once the target is captured, another antibody labeled with the fluorescent tag attaches to it like a flag. For some antibodies labeled with biotin, a fluorescent helper called PE streptavidin is added to make the signal stronger. Because each bead has a unique color or the unique code, the system is able to detect many different molecules at the same time. This lets you measure multiple targets in a single sample with high sensitivity and give you accurate results. Okay, this kit is truly versatile and can be used with a wide variety of samples, including serum, EDTA plasma, cell culture supernatant, and other biological fluids. For all the sample types, the key is to collect the liquid portion carefully and remove any cells or debris by centrifugation. This is the only step where you will need the centrifugation for preparing the sample. After that, you won't be needing any centrifugation machine. This ensures that assay measures only the soluble targets and gives accurate reproducible results. By supporting multiple samples, the kit also provides flexibility for different research applications and experimental designs. Okay, now the operational procedure. This kit also follows a really straightforward workflow designed for efficiency, reproducibility, and ease of use. First of all, the lyophilized standard is reconstituted and serially diluted to generate a standard curve for quantification. Samples and standards are then incubated in a pre-mixed antibody conjugated beads, allowing the target molecules to bind specifically to their specific beads. Detection antibodies are added next to label the capture target, followed by SAPE, streptavidin, and PE incubation to generate a fluorescent signal. After the final washes, the beads are resuspended to read on the flow cytometer. The assay is flexible, incubation times and volumes can be adjusted depending upon the sample type or equipment. Overall, this workflow enables simultaneous, highly sensitive measurement of multiple targets in a single sample while conserving the time and materials. Okay, let me give you a brief idea how you have to set up the experiment and what each population means. Sample after the assay, samples can be analyzed by flow cytometry. First of all, what you will look at the data, it will look like something like this. Firstly, this is the main bead population, which is P1 or R1 here. You can also label it as P1. This is the first gate using forward scattered and light scattered channels. Each bead set is then distinguished by its unique fluorescent code in the APC or APCCY 7 channels, which tells us which target it represents. For example, here you can see IL-6 is representing 25.55% percentage of this total population. IL-2 is representing 22.61% of this population. So here, this is the whole population, and these are the four targets we have analyzed, we have captured and segregated for our analysis. The PE channel is actually used to measure the fluorescent signal for each target, and same gates are applied across all samples. So it is very, very simple process. Each sample gates are checked to ensure the accuracy. It is also a very important step. Finally, we can move on to the statistical data. We can export it, including the target names, bead counts, and median fluorescence intensity, providing a quantitative readout for each analyte. Before generating the standard curve, the data have to be processed by taking the logarithm of both the mean fluorescence intensity values and the known concentration of the standard. These values are then transferred into the statistical software, such as we recommend Origin software for the statistical analysis of antibody antigen interactions, because this software has the 4-parameter logistic curve function, which is the most suitable for the antigen antibody reaction. Other than that, you can also use SPSS or GraphPad Prism. We do not recommend Excel because Excel doesn't have the 4-parameter logistic curve function. Now here, once the values are transferred into the statistical software, we have to generate the standard curve. The resulting equation from this curve is used to calculate the concentration of cytokines or other targets in each sample. This ensures that the measurements are accurate and comparable across different samples, providing reliable qualitative and quantitative results for your research. Okay, the AptPlex kit also demonstrates a robust and reliable performance. The detection range covers 5 to 5,000 picogram per ml for most cytokines, with CXCL10 and CCL2 starting from 10 picogram per ml, providing a broad linear range for accurate quantification. The limit of blank for all analytes is less than equal to 8 picogram per ml, ensuring sensitivity for low abundance targets. Recovery is also excellent, ranging from 70 to 120, and precision is high with intra and interassay variation under 15%. More importantly, the kit also shows strong specificity with no significant cross-reactivity among the included analytes. These performance matrices confirm that AptPlex delivers reliable, reproducible, and high-quality multiplex measurements for research applications. Now, just move on to the next and the last section, the advantages and the competitive analysis. Okay, what really stands out here are the practical advantages for researchers. You can measure over 30 indicators in a single tube, which saves a lot of time and avoids any variability from running multiple experiments. It also works with a wide variety of samples and very small sample volume, just 20 microliters. So it is ideal when the samples are limited. The workflow is quite efficient with a total reaction time of about 2.5 hours, faster than many comparable methods. Sensitivity is strong with the detection limits below 5 picograms per ml, making it suitable for low abundance targets. Another key point is the compatibility. It works with most standard flow cytometers, so there is no need for the specialized equipment. And finally, the flexible and customized panels also allow researchers to design experiments based on their specific needs. When we compare AptPlex with BD Biosciences, AptPlex runs a full assay in just 2.5 hours compared to 3.5 to 4 hours on BD instrument. Sample volumes requirements are also low, only 10 microliter of blood or 20 microliter of supernatants. Magnetic bead separation gives full flexibility in the workflow, and it's also cost competitive while supporting up to 30 analytes per panel. Delivery is also faster, as we can deliver this kit in just 1 to 3 weeks. So in short, it combines speed, flexibility, and low sample use and multiplexing capacity. Okay, a similar analysis is along with the BioLegend. Then many people ask me the differences between AptPlex and Luminix. Actually, they both use the same base-based multiplex amino acids, but they differ mainly in how the system is implemented in the lab. AptPlex is designed to run on the standard flow cytometer, which means that most labs can use their existing instruments without needing a dedicated multiplex reader. Luminex require a separate proprietary analyzer built specifically for its own bead detection system. Because AptPlex uses flow cytometry for readout, it fits directly into the labs that are already performing flow cytometry, allowing a multiple data generation within the established workflow. Luminex also operates as a standalone platform with its own dedicated instrument, its own software ecosystem. Bead identification is also handled differently. In AptPlex, bead populations are distinguished through flow cytometer signal detection, while Luminex use internal fluorescent dye ratios that are interpreted by its own dedicated instrument. So for the practical laboratory perspective, AptPlex reduces the need for additional equipment, investment, and integrates into existing setups where Luminex requires adoption of a complete system specifically built for that particular platform. So that's how easy you can use AptPlex. Okay, now take a look at all these targets this panel covers. You have got the main T helper, T helper cells like Th1, Th2, Th17, plus some CD8 positive T cell cytokines, giving a clear picture of immune activity. Chemokines are also included, which helps track how cells move and communicate. B cells and growth factors are also covered. So you can study the immune regulation, cell growth, and tissue repair. And it doesn't stop there. These panels reach into inflammation, stem cells, macrophages, tumor biology, and even new T cell subsets like Th9 and Th22. All of this together means that you can see the bigger picture and understand complex immune responses in one go. AptPlex isn't another essay. It's a research tool designed to unlock deeper insights into the complex biological systems. By combining a broad target coverage, high sensitivity, and flexible, efficient workflows, it empowers scientists to ask bigger questions and get answers faster. Whether it's exploring immune responses, inflammation, tumor biology, or novel pathways, this platform gives researchers the confidence to pursue advanced studies without compromise. In short, AptPlex empower labs to explore immune signaling, inflammation, tumor biology, and beyond, all on one flexible, efficient, and sensitive platform. It's about making research faster, smarter, and more insightful. That's all for today's lecture. If you have any questions, please write them in the chat box.